that PCR is simple, it's fast, and it is one of those reliable tools. And we all know that the PCR has played a very, very significant role in the current pandemic, right? So even before the current pandemic was on the, horizon, PCR is being used for many different purposes, right? Typically, for example, when you wanted to know whether a person who is on anti-HIV medication, if they wanted to know, what is the viral load in such people, they would typically go for PCR, real-time PCR, to know how many viral particles are there. Okay, so it is something not new, but it is just that it has been adapted on a global scale with the COVID pandemic, real-time PCR. So we will start off from where where we left last time, and then we will start looking at the real-time PCR in today's class, and then we will also delve into a few other things, very very interesting things. Let me share my screen first. Mm. My laptop has a mind of its own, so sometimes it behaves a little differently. Okay, there it is. All right, there we go. Are you able to see this? Okay, so traditional PCRs are typically called as endpoint PCRs. What is the endpoint? We want to know at the end of 25 cycles or 30 cycles, did we get the PCR amplification right or not? Was it a success? Is it an S or no? In this case, you can see that lane 1, 2, and 4 and 5, they have positive amplification, whereas lane 3 is a failed PCR, right? So this is typically 25 cycles, depending on different machines. It would take anywhere between 2 to 3 hours, and then at the end of 2 hours, another maybe half an hour or so to run a gel to check whether you have a PCR product or not. 3 hours, right? So that's probably was okay in the old days, when the technology was still in its infancy. But today technologies have matured so fast that 3 hours seems to be a long time. So people have started looking to see, is there a way that we could, visualize this even before, instead of waiting till 25 cycles and then loading your gel and then running an agarose gel to visualize, is it possible for us to visualize this reaction, as it goes? As you can imagine, let me go back a couple of slides. Um, this one, right? So as the reaction progresses, you can actually see, from 64 to 128, to as the number increases, there has to be a way. There has to be a way that we could quantify the increase. There is an exponential increase, and if there is a way to quantify this increase, that would be great. Okay, so that is what we are going to look in today's class. Okay, let's go back to this slide. So this is what we were talking in the last class. Bio-Rad is one of those commercial organizations, that makes these PCR machines, makes reagents. It's not only Bio-Rad. There are several many different companies. It is just that they had this nice handout education material, which I thought was very very useful. So I'm going to use that. So thanks to Bio-Rad for doing such a wonderful job. Okay, so this is the same picture. If you look at it, lane 3 has failed. Lane 1, 2, 4, and 5 has amplification. And the question is always, if you see lane 1 and lane 2, there is a single PCR product. Lane 4, you can actually see there is probably some kind of a contaminant. Lane 5 has multiple PCR products. The question is, what are you looking for? Are you looking for a single product or are you looking for multiple products? Okay. There are certain enzymes, there are certain genes that are that constitute what is called as a gene family, right? So sometimes when you do a PCR for such gene families, you may inadvertently, amplify multiple copies. Okay. So there are principles for designing your primer so that your primer, aligns or, goes and binds to only select regions so that you can have specific amplification. There are times when you may want to know how many different kinds of family members are there. Then you could use certain special oligos, so that will give you a rough estimate of how many such gene families could be there. Nevertheless, so this, as I said, is an endpoint PCR. You have to wait for your 2 hours or 2.5 hours, maybe 3 hours, to know the result of your PCR reaction. And if it has failed, you will have to redo the thing all over again, right? So instead, you know, so how does it work? We all know that there is a double stranded DNA. It has two primers, one which is called as the forward primer and one that is called as a reverse primer. Okay. Then you have lots of DNTPs and you have a thermostable DNA polymerase. It could be Taq polymerase. There are many other polymerases. Okay, in a buffer, and then you have a denaturation, then you have annealing. This is something that we all know very well, and then TAC extends it, and then this entire process is repeated until you have multiple copies of it. Okay, so that is how traditionally PCR is usually represented. Okay, in order to understand real-time PCR, let's simply use certain numbers and some thought experiments. Okay, and this the number, you remember I said that the amount of amplification is simply given by a very, very simple mathematical formula, which is 2 raised to the power n, where n is the number of cycles. The number of cycle could be 25 cycles or 30 cycles, and depending on whether it is n is 25 or 30, the copies are going to be extremely different. There is an exponential increase. So we're going to simply look at this extremely very powerful and intuitive this thing, and then we'll do a series of back calculations to understand how can we visualize. Not only visualize, you are in essence you are going to quantify using real-time PCR. Okay, are we are all on the same page? Can we move ahead?
Sir, it's clear.
Okay, thank you. Okay. So to understand real-time PCR, let's imagine that we are in the cycle number 25. Okay, PCR cycle reaction number 25. Okay, and so in our this thing, the PCR tube, you will have nucleotides, you will have primers, forward primer, reverse primers, right? You will have the template DNA, and then you will have all these different, you have amplified till 24th cycle, right? All those PCR products will be there. You have Taq polymerase and all that. Approximately, you may have about 1 million copies of the amplicon at this point in time. Approximately, this is just a calculation, rough calculation for our this thing. Okay, so this was at 25th cycle. You have 100,000 copies. Okay, as I said, in this one, the number is different. Okay. But for our mathematical calculation, we are taking it as 100,000, 1 million copies. Becomes easier. Okay. It could be different. It is definitely not 100,000. It is going to be much higher. But I'm just using this, 1 million as a ballpark figure so that you can have easier calculation. So if it is 1 million in 25th cycle, what would be there in the 24th cycle? It is exactly half of what it is in the 25th cycle, because as we know, 2 becomes 4, 4 becomes 8, 8 becomes 16, right? So what was there before 16? It was 8. What was there before 8? It was 4, right? So if it is 1 million, then it is likely that in cycle 24, you're going to have about 500,000 copies of the amplicon. And what will happen in cycle 23? You will have about 250,000 copies of the amplicon. And in cycle 22, you will have 125,000 copies of the amplicon. So you can actually see each of them doubling, right? From 22 to 23 to 24 to 25, you are having an increase in the amplicon from 125,000 to 250,000 to 500,000 to 1 million copies. Is that clear?
Yes.
Okay. Very good, very good. So if we were to graph these numbers, if we were to put it on a graph, okay, then typically you will see a graph somewhat like this. Okay, it is slowly starting to increase. It is part of a sigmoidal graph, right? I mean, I'm sure all of you would have done, Excel sheets, right? And what you're going to see is something like a sigmoidal graph. So this at the bottom axis represents the cycle number, right? 24th cycle, 25th cycle, 22nd cycle, whatever. So you're going to start right from the first cycle till your 25th or 30th cycle. Okay. What you see on the x-axis is the cycle number. What you see on the y-axis is the fluorescence units. We'll come back and see how we get this fluorescence units. Okay, we'll just wait for a couple of slides. You will know that. Okay, so we're measuring two different things. One is the cycle number, and one is the intensity. Okay, or the fluorescence intensity of the amplified PCR products. Okay, can we go ahead?
Yes, sir.
Okay.
Yes.
So at 25th cycle, okay, in our Eppendorf tube or near PCR mix, there are about 100,000 or 1 million copies. Okay. And so what will happen when it is in cycle 26? Any idea? If it is 1 million copies in cycle 25, how many will be there in cycle 26?
2 million.
Very good. Very good. 2 million. And what will be in cycle 27?
4 million.
Very good. Suppose instead of, cycle 27, 28, suppose we do say 50 cycles. What will happen? So as the cycle number increases, okay, the primers get consumed. Tag polymerase is stable, but the primers gets consumed. The DNTPs get consumed. So at some point, there is no longer an exponential increase. Got it? You cannot have exponential increase forever. Otherwise people would do PCR for probably 2-3 days together, and they might that is not possible, right? Chemically, biologically, and in every other sense doesn't make sense. Is that right? So this reaction has to reach a plateau, right? So you can't have these many amplicons. I mean, it's like a absurd number. So typically, it doesn't happen. Okay. So exponential growth does not go on and on. Okay. So typically, around some this thing, depending on how what your starting point is and all that, around 30th cycle or little later or little earlier, you will start seeing a plateau. Okay, you will start seeing a plateau like this. So there is what you actually see, as I said, is a sigmoidal graph. Okay, you will see barely any signal, and then there is an exponential part, and then it becomes a plateau. Okay. So now we are going to put our numbers now. Okay. Now, how do we use all this to measure? Simple. So let's imagine two people are doing experiments. Okay. And let us say one person starts with 4 times the amount of DNA. Say it's just a pipetting error. Okay. Instead of pipetting something, this person or the student pipetted it 4 times extra. Everything else remains the same. Everything else remains the same, except that the person pipetted the template DNA 4 times. That means your initial template concentration is 4 times higher compared to the other student who did perfectly well. He or she was asked to do it this way. They did exactly the same. The other student, by mistake, committed an error, in pipetting the template DNA. So let's just say what happens in cycle 23. Because at cycle 23, as we saw in our previous calculation, you're supposed to have 250,000. Cycle 24, you will have 500,000. Cycle 25, you will have 1 million. Okay, because you started with 4 times higher template. Okay, that means you are going to have amplification at a much, much faster rate. That means by cycle 23, this is just a theoretical example. This is just a theoretical example. By cycle 23, you may have about 1 million. By cycle 24, you will have about 2 million. And by cycle 25, you will have about 4 million. Okay, so this is the numbers, assuming that, you have done, something 4 times more template. Imagine this situation. You are going into a COVID hospital and you have a patient who is in advanced stage of the disease. That means his viral load is going to be much higher. Okay. And then you have another patient who is just showing the beginning of the disease. That means his or her viral load is going to be much, much lower. You can simply compare it that way. One person having an initial stage viral load is very low, one person having a severe disease where the viral load is higher, and you're trying to detect the presence of virus in the sample. Does that make sense?
Yes, sir.
Okay, very good, very good. Okay. Okay, so if you were to plot a graph of these two, the green and the red, right? So this is the green and the red. If you were to plot a similar graph on the Excel sheet, what you will see is, you will see a graph that looks very similar. You will still have a sigmoidal graph. You will still have a stationary phase, but then the green guy would probably jump, this thing by a few cycles ahead, right? Because as we saw, the green thing has 4 times more samples. So, or you can simply say this could be the person with the severe disease, and this could be the person with a mild disease, right? Okay, so this is how the plot looks like. Okay. Okay. So if suppose, you start with this thing, less number of this thing samples, okay, less number of the template, then typically this number gets reversed. Okay. In this case, we are showing you a sample where, when you have 100,000 in cycle 25, this person, because of the pipetting error, this person has only 125,000, cycle 26 has 250,000, and cycle 27 has 500,000, and cycle 28 has 1 million, right? So as again said, you could correlate between doing the experiment in the lab, or you could simply imagine a situation in the current pandemic where you have people with different COVID viral load. Some people have extremely high viral load. Some people may have a bare minimal viral load. Some people may have intermediate, anywhere between these two extremes, right? And you are going to do a real-time PCR for maybe 100 different patients, and all these 100 different patients can have very different viral loads, right? Does that make this thing sense? So we saw one in which there was too much of template, and in this case we are seeing too little of a template. Okay. Can we go ahead?
Yes, sir.
Okay. So if you were to plot this graph, beautiful, isn't this beautiful? That you are now able to see 3 different lines. They all resemble each other. They all resemble each other. Very beautiful sigmoidal graph, right? And so this is the one that was normal. This is the one that had extra template, and this is the one that has less number of templates. Simply put, let us say that this is green is the person who has a very severe COVID disease. Okay. And this is a person who has a moderate disease, and this is the person who is just beginning to show the symptom of the disease. So you can imagine, if you take 100 patients, and if you do a real-time PCR, you are going to have multiple graphs overlying on each other. right? Okay. Okay, so in order to make sense, people simply use a graph. This graph, if you actually go back and see this, okay, if you go back to the previous graph, the this thing, if you were to blow up this region, the lowermost region, it look very different, okay. But we are not going to go into discussion, except that we will say that this detection, it generally uses a thing called CT value. Okay, so I'm sure all of you must have heard about CT value in the real-time PCR when we do our corona test, right? So CT value is nothing but cycle threshold. What is a cycle threshold? A cycle threshold is defined as the number of cycles or number of amplification cycles required for the fluorescent signal to cross a certain threshold, before it exceeds the background level. Okay, so all these real-time PCR chemistries are done where there is a lot of background signal, and you want to have this CT value which is way beyond those background signals, and where it clearly establishes that the amplification is starting to take place. Okay, so that is what is called as the threshold cycle. The one beauty thing of this threshold cycle is that when you have excess template, you will see that the threshold is reached at the 23rd cycle. When you have a normal template, you realize that your threshold is reached at 25th cycle. And when you have less template, the threshold is reached at 28th cycle. So this CT number to an extent gives you the copy number or the relative proportions, right? So this is going to be, maybe 4 times higher, and this is going to be 4 to 8 times lower. Okay. As I said, these are all calculations to show you, to make you understand that it is possible to quantify the copy numbers, or the viral loads, or to quantify the actual amplicons. Okay, so this CT value is an arbitrary value. Okay, in the early days, people would set their own arbitrary values, but nowadays the machine comes with very, very advanced algorithms where it automatically sets a threshold value, and all you need to do is set up the PCR reaction, and it will start giving out the values very quickly. Okay. So is this thing on threshold value, did you all understand?
Yes, sir.
Yes, sir.
Okay. So the quantity or the amount of this thing is simply, given by a mathematical formula which says it is 2 raised to the power CT. Okay, so that actually is, you can do the quantification. Okay, we are not going to go deep into this because we will probably do this in the later this thing. So just quickly, this recaps, we have 3 different reactions, one in which you have 4 times. So this is the normal one. The red is the normal one, normal PCR. In one, you have 4 times excess template DNA, and in another one, you have 1/8 of the template DNA. And when you do a PCR reaction, real-time PCR reaction, this is the graph that you obtain. Okay. So the biggest question right now is, what is this? How are we detecting this? Right? That is what we are going to do now. Okay. Okay. So there is a direct relationship between the starting amount of DNA and the cycle number, and that you will reach an arbitrary number of DNA copies. Okay, that's what I said. It's given by this formula, 2 raised to the power cycle number. Okay. And so this real-time PCR is extremely sensitive, okay? And theoretically, you should be able to detect even when there is a single copy. But because of the detection limits, because of the laws of physics, generally, you would see about 100 copies is the minimum amount. But then nowadays people have broken this. They have taken multiple approaches, and now you can actually talk about few particles. Typically when it comes to HIV, they're able to see, they're able to give as low as 10 to 20 viral particles. So they have gone down. Okay. So it is much more advanced. So how do we measure? How do we measure this DNA? Typically, when you run a gel, you can visualize it and say yes, there is a DNA, right? Or you can simply take it to the spectrophotometer and read absorbance at 260, and 260 all these purine and pyrimidine bases, they get excited. So it's a direct proportion, and you will be able to quantify the DNA, right? But in real-time PCR, how do we actually measure the DNA? There has to be something different. We can't be, at the end of every cycle, we can't be taking it to the spectrophotometer, right? So two dyes, that were, so typically we use ethidium bromide when we run agarose gel, right? And ethidium bromide cannot be used in the laboratories for diagnosis because it has a lot of different kinds of, disadvantages. So ethidium bromide is generally not used, okay? People started using another dye called cyber green. Okay, so cyber green is very much like ethidium bromide, but it is less carcinogenic, less mutagenic, and it gets degraded in the environment very easily. Ethidium bromide is carcinogenic. It could be toxic. It has the potential to cause cancer. So people have started, minimizing the use of the ethidium bromide. They've started using cyber green. Okay. Now, whether you use cyber green or ethidium bromide, the way you are able to see the DNA is because it intercalates between the double stranded bases, and then when you shine UV light or appropriate light, it fluoresces. Right. That is how you are able to visualize the DNA in the agarose gel. Okay, so cyber green dye fluoresces much more brightly than ethidium bromide. Ethidium bromide is still commonly used. In fact, in our university, in our classrooms, we still use ethidium bromide. Okay, but the way it used to be used earlier, is very different. Nowadays, we have use extremely tiny amounts of the DNA, tiny amounts of ethidium bromide to visualize it. Okay, which is fairly effective. Okay, but it is not possible to use ethidium bromide for quantifying in a real-time PCR environment. Okay. So people started using cyber green. Okay. And one of the so this is the ethidium bromide. It basically goes and intercalates into the double stranded DNA, and it fluoresces. It fluoresces under UV light. Okay. Whether it is bound to DNA or not, ethidium bromide also has a background fluorescence. So it will keep fluorescing even when it is not bound to DNA. Okay, so that is a disadvantage. Okay, so this is the cyber green dye. Okay, this is a double helical DNA, and this is the one. This is cyber green, which is bound to the major groove of the DNA. Okay, and you can actually see that it also fluoresces. Okay, when it is bound to double stranded DNA, it also fluoresces. And more importantly, if you look at cyber green, it is not at all mutagenic. Okay, it is not at all mutagenic. Okay, there is so this is a dye that is extremely safe to use, safe to handle. Okay, so why do we still use ethidium bromide? Simply because ethidium bromide is much cheaper compared to cyber green. But although these days cyber green reagents have become much cheaper than what used to be earlier. Okay, so people are in the process of shifting from ethidium bromide to cyber green slowly. Okay, can we move ahead? Okay. So this is the dyes that are used in real-time PCR. Okay. So as one of the property of cyber green is that it binds to double-stranded DNA. Okay. It doesn't bind to single-stranded DNA. It binds preferentially to double-stranded DNA. So at the end of say 25 cycles, when you have about 1 million copies of the DNA. Okay, of the PCR product, cybergreen is going to bind much more strongly, right, compared to, say, in the 10th cycle or maybe even earlier, right? So as the number of double-stranded DNA keeps increasing, you will see that the cybergreen also keeps increasing in its fluorescence intensity. So basically, at the end of this cycle, okay, you can quantify this simply by exciting with certain, laser pulses, okay, and then the cybergreen dye will absorb that radiation and then it emit at a longer wavelength, which can be detected by the probes in this machine. Okay. Um, okay. So there are many different dyes that are there. Okay. Typically, cybergreen is just one of the dye. There are many different dyes, and each of them, comes with its own advantage and disadvantages. Okay. So there are three components, main components in a real-time PCR. You have a thermal cycler, you have an optical module that will detect fluorescence, as the reaction is progressing, and then you have a computer attached to it, which will translate the fluorescence data into the plot that you're seeing in the Excel sheet. Okay. So quickly, this is just an example. This of course is very old time real time PCR. Okay, this model I'm not sure whether they are still having it, but this used to be called as Bio-Rad IQ5 real time PCR experiment. This is just showing you that it fires a laser pulse. The laser pulse is directed towards each of these 96 well plates, and then each of these 96 well plates in turn will emit radiation, which again is collected and it gets into the photomultiplier tube, which registers the intensity, and then it starts plotting the graph for us. Okay. Okay, this is just different versions. They are showing you the internal parts of the machine. Okay. Okay, so essentially, it reads all these different wells, okay, and then each well corresponds to a data point, and then it plots. Okay, so that means you can run literally 96 real-time PCR experiments in one stretch. Okay, but generally people don't do 96 at a stretch because you always have certain controls as positive controls. We'll also have certain controls as negative controls, so that you will know what is the background noise that is caused by these different dyes. As I said, cybergreen is just one dye. There are several other dyes, that are used in real-time PCR experiments. It depends on the kit. So somebody may be buying it from Bio-Rad, somebody may be buying it from Thermo Fisher, somebody may be buying it from some other company. Each company's this thing kit will have a different chemistry associated with it. Okay, so the dye chemistry is extremely, it is patented and it is generally, very specific. Okay. Okay, how does this real-time PCR data look like? So from a real-time PCR, you will see a very different graph. So this is the cycle number ranging from 0 to people go up to 40 cycles, but it is not necessary. Okay, and then you have these different fluorescence intensities. Okay, and if you see this, if you transform this data into a logarithmic view, then you will see a graph somewhat like this. This is the actual data in the logarithmic view. And then you actually have so this is all the background noise. This is all the background noise that you're seeing this in this thing. There are 96 reactions that are going on. Okay. And so you have a huge background. But then among this background, as the reaction progresses, you can actually see that the signals are becoming more and more specific. That is why, you set up a CT value or cycle threshold beyond which this amplification is taken into consideration. Okay. And this, as I said, initially people used to do it manually, but nowadays You have sophisticated algorithms that can detect what the background noise is, and accordingly, they will, decide on the CT value. Okay. Okay. So, as we can see, the fluorescence data collected during PCR tells us how much, is it, 4 times more, or is it like 8 times less? Okay. But there's another type of analysis we can do that tell us, what actually is happening on in the real-time PCR. Okay. This is what is called as the melt curve, concept of melt curve. Okay. Simply as I said, the cyber green dye or any of these dyes, they have the propensity to bind to double stranded DNA rather than to a single stranded DNA. So when it is, when the two strands are separated, they are not going to bind. During the process of annealing, when the primers are annealing, when you see a double stranded DNA getting formed and it is getting extended, you will see that the dyes will start to bind. Okay. And then when it is as the reaction becomes cold, or in the this thing, when you're trying to do annealing and extension, when you will see a significant amount of double stranded DNA, you will see that the proportion of double-stranded DNA molecules is much more. So dyes, the amount in the quantity of the dye that goes going to bind to these double-stranded DNA is also equally more. Okay, so you can actually simply see that you can visualize this. You can visualize this in this thing. Okay, so melt curves are tells us how many products are there in the reaction. As I said, one it is one thing to have 96 samples, okay, but then the question is, among these 96 samples, if you want to categorize them into mild, viral load, medium viral load, normal viral load, or extremely high viral load, this is what is going to help us differentiate that. I don't think we are going to spend considerable time in this, okay, because this is like beyond the scope of this class. We are not going to spend some this thing. Instead, we will focus on using real-time PCR for diagnostics. Okay. Maybe not in this class. We will do this in the next class. Okay. How can we use this, in the actual crime scene, and what is the basis for using in the crime scene? We will have a quick look at it in the next class. Okay. So any questions in today's class? I will share this, maybe in the next class. Okay. The slides. All right. Okay. Any questions on this?
Sir, I have a question.
Yes, please.
Sir, what is internal control in RT-PCR that we use, like in the case of detection of
Very
COVID?
good, very good. Internal control is a positive sample, something that that has a viral, known amount of viral DNA. Okay, so that you can say, so that is the normal viral load, or the person that is infected with the normal amount of this thing. You can have people on either extreme. Either you can have people with a less viral load, or people with a maximum viral load. So that is used as an indicator. Okay. It is something like, say, in a class, in your class, for example, in your class of 5th semester, suppose we say, what is the average CGPA, right? You have people above the average CGPA, and you also have people below the average CGPA, right? So it's a broad range. So this average CGPA, is set as a benchmark, and then you start realizing that you have people above CGPA and you have people below CGPA. So this internal control is somewhat like that. You have, so this will also tell you whether the reaction, PCR reaction, has worked well or not. Okay. If it has not worked, then it will tell you that this reaction, this result cannot be taken into consideration.
Sir, also one thing,
Hmm.
while I was performing RT-PCR,
Hmm.
the graph of internal control was not like it was linear, but
Hmm.
still we get the CT value. How is it possible?
It was linear. You are saying your positive control was linear.
Yes, sir. Not positive control, but the IC value, IC graph in every sample.
Okay. No,
Still,
that
we
must
get the
be.
CT value.
I'm not sure. I'm not sure. I'm just guessing. This could be a mathematical transformation of something. I'm not sure. I'm not sure. I'm just giving it a guess. Okay. This could be some kind of a mathematical transformation by the software. And then that's what I said. Earlier people would do this by themselves, but nowadays most of this has been taken over by all these softwares that transforms the graph and then it calculates based on certain computational programs. I'm not aware of it. I'm not aware of it.
Okay, sir.
Okay. Any other question?
No.
Okay. Okay. So if not, then what we will do is we will come back. We will look at this because I want to make sure that we are going to look at parts of our own genome. Very short this thing presentation on how the human genome has certain unusual properties and how those properties can actually be used for our DNA fingerprinting. And that is typically what happens in a crime scene. People go and based on the hair samples, based on the blood samples, or based on any other biological sample that may be there in a crime scene, people are able to now go back and trace out who the person could be. So we will probably look at it in our next class. All right. Okay. So if there are no questions, then we'll meet on Monday then. All right.
Thank you, sir.
Okay. Take care.